Professor Veronika Magdanz joined Systems Design Engineering Department at the University of Waterloo as an assistant professor in 2022. Following her PhD in biology from the University of Dresden on the development of sperm-driven microrobots, she joined the Technical University of Dresden to continue her research on the metabolism and kinetics of sperm cells. She was later awarded the Humboldt Fellowship at the Institute for Bioengineering of Catalonia, where she explored medical applications of magnetic field-guided small-scale robots. The first time I have heard of Professor Magdanz's work was in a seminar she gave to the VIPlab group here in this very room last year. I remember sitting there and feeling my jaw progressively drop on each slide in the presentation. Indeed, microrobots made of sperm cells, which are guided by magnetic fields, might sound like something straight out of science fiction when you first hear about them. Professor Magdanz is one of the leading researchers of this very much real technology, a technology which not only sounds cool, but more importantly, has a surprising number of clinically relevant applications with the potential to help people. Professor Magdanz, we are honored to have you here, and we're extremely happy that you have found time to talk to us.
Thank you so much for having me.
Yeah, so usually we start with asking directly about research, and that's what we've done for the last few episodes. But right now, the topic of research that you work on is very niche. So the most pressing question that I have on my mind, that we have on our mind right now, is where did this all start? At what point did young Veronika make the decision of devoting her life to microrobotics?
Good question. Yeah, I don't think I ever made that choice actively. I think that just sort of happened, and I followed that path of opportunities. But specifically, how I ended up working in this research, let's say, is so I studied biotechnology, as you mentioned, and then I was always motivated going in that direction because I wanted to learn about nature and use that knowledge to develop new technologies, specifically to improve health technologies and find solutions for all these global issues we have in health. So after I took a bit of a break after finishing my degree, my diploma, which is kind of similar to a master's degree, I took a break, and at some point I thought I was ready to jump back into research. I was motivated and started looking for positions, for PhD positions, and I saw this post that was I think it was on Nature Jobs. So it was like a global posting for PhD position in Dresden at the Leibniz Institute for materials research and solid state. And it made me very curious because it said something like, so the posting was finding or using. It was a very strange title because I think it said something like using bioorganisms for microrobotics. And first I thought, as a biotechnologist, this bioorganisms, what does this even mean? Like, of course, organisms are biological. But then I read a posting, and it was about microrobotics, micro-nanorobotics. And this group in Dresden was one of the few worldwide at that time fabricating self-propelled small devices that are able to deliver drugs, deliver cells, manipulate cells, and so on. So I'd never heard of that before. And I thought, oh, that sounds super interesting. So I decided to put an application in, knowing that I'll probably have no chance because this is posted globally on Nature Jobs. I haven't been working in academia for the last 2 or 3 years. But anyways, I'll do it. So I put my application in, and indeed I got contacted by the PIs, and we had an interview. And so they explained to me that they're looking for a PhD student that would look into finding better biocompatible solutions for propelling microrobots. So they shared with me that they had some work on moving them with chemical reactions or moving them with magnetic fields, but they wanted to expand on having really a biocompatible solution to move it forward towards medical applications. And again, I didn't really know much about it at that time because I was just applying for the position. But I said, okay, that sounds really interesting. I'd love to work on that. And indeed, in the end, they offered me the position. So I was in Canada at that time. So I moved back to Germany. I moved to Dresden to start this position. And the first few weeks, I was reading a lot about microrobotics and different strategies, how we could propel them with more biological mechanisms, using enzymes, maybe using magnetic fields, using bacteria, using yeast cells or reactions of yeast cells that maybe produce gases and so on. And I remember this very awkward meeting with my PI about 3 or 4 weeks in, where I showed him, my ideas, what we could do, maybe using this enzyme or that. And I remember getting very nervous, and you can imagine, like, I was maybe, in my mid-20s, and there were mostly men working in this group. And this young group leader, he got very nervous, but kind of said, we've been talking about this, tossing this idea around, and I know we haven't told you yet, but we would really like you to explore if you can use sperm cells to propel these microtubes. So at that time, we were using rolled up nanotechnology to make small microtubes or nanotubes for all kinds of applications, for nanobatteries, for resonators, microrobots, and so on. So he got quite nervous because it was a material science institute. We had no biolab. They had no idea how to work with cells, where to get sperm cells from, and obviously that was a little strange. So he suggested that idea, just tossed it out there, and I thought, sperm cells, okay. So the idea was to put these sperm cells into these tailored microtubes and then use the sperm cell as propulsion source, and then use magnetic fields to guide them in certain direction. And so I said, well, okay, I can try it out. Where do I get sperm cells from? And he just said, well, you figure it out, like, you're the PhD student. so anyways, I've always been never afraid to try new things or to explore unconventional ways, let's say. And okay, think about where do we get sperm cells from without making it completely embarrassing but for anyone. But I remembered because I grew up on a farm, we were doing artificial insemination in animals all the time, in horses, in cows. So I said, well, there must be farms around where I can just get some samples of bull semen without, killing any animals. Usually bulls is an easy animal to work with. And so I made some calls, and sure enough, they were happy to give us bull semen for research from a local semen export company. And I remember my first experiment, just putting the motile sperm cells together with these microtubes that I had designed to sort of fit single sperms, and the sperm cells would just swim into these microtubes and start carrying them around. And it was just really amazing seeing that under the microscope. And that was sort of the start of my PhD journey, and start of that actually new field, you could almost say, of biohybrid microrobots, of integrating sperm cells with artificial components for new applications, either to propel things, but also for drug delivery, for cell delivery, for diagnostics of sperm, and so on. So there was a really exciting new start. And from there, it was just, I almost want to say it was easy, because it was a completely untouched field, right? Nobody had ever done anything similar. So it was very easy to get the first papers out, to get excitement about it and interest. And so that's how it all started.
That's amazing. So you've started out with this in Dresden, right?
Yes.
With this journey of microrobotics, but then you went to Spain for a little bit. Is that correct? So you have spent some time in Barcelona, and you're reportedly working also on stuff like 3D printing of muscular tissue. So did you decide to switch fields temporarily to explore, or was it also somewhat in concordance with what you were planning to do afterwards?
Yeah, good question. Sometimes there's discrepancy about what you think you should do next, and or what you want to do next, and what the opportunities are. That's maybe one thing at the side. But so I did my PhD on the microrobotics, working with sperm cell, not really knowing much about sperm cells. So we used them because they were motile. We knew how to thaw them and handle them, but that was about it. So in my first postdoc, I said, I want to learn more about biology of sperm. Really, how do they move? Why does that one cell make it to the egg and not the other millions? Like, what's special about it? Is it just the most motile one? Is it the one that can manage the energy the best? And those kind of questions. So that's what I did in my first postdoc. And then I applied for the Humboldt Fellowship to go to Barcelona with a project that actually never worked out. So thankfully, this Humboldt Fellowship is given to the person, not to the project. I say for the project. So halfway through, I actually changed. So initially, my proposal for the Humboldt Fellowship was to develop insect muscle-based actuators, because insect cells are able to regenerate and self-repair, which is a really unique feature that we can't really engineer very well. And that was during the pandemic, so we ran into a lot of trouble even getting the cells. I remember getting them stuck in customs 2-3 times, spending a huge amount of money on that, and we were never able to get these insect muscle cells going in culture. So at some point, I decided with my supervisor in Barcelona, we're going to drop that. And his group in Barcelona, one of the areas was muscle actuators. And so I joined that team to learn new methods. That was another goal. So, of course, I wanted to develop my own research profile, but also learn, gain new skills. So that's, I think, was what my goal was for the postdoc. So I kind of joined that team on 3D printing muscle tissue. So that's still not published. We're still finishing that up. But that was really neat to learn how to 3D print biological tissue. And there we're still at the very beginning. So it's really just one cell type that we're printing right now. But we get it fully functional. So they really contract, they differentiate it, they show good contraction and good muscle performance. So so that's one of the things I added during my postdoc.
I see. So did this experience actually help you in your main research area? Because I believe that a project either done, or if it's not even fully completed, even if it's partially done, does lead you to have a set of observations. And it needn't be published, in my viewpoint, to learn something out of it. So did you have that learning curve in this project? And were you actually able to apply that into your current research with sperm microrobots?
Mm-hmm. Yeah. I would almost disagree with you there and say, even if you haven't published it, you learn a lot. Sorry. So I've learned a lot, especially in terms of skills and like lab skills, and because there's a lot of experience that you need for the 3D printing. It's not an easy protocol to follow. How does that fit into our current research is that we're now exploring to create in vitro organ models. And these can be very simple, This can be maybe just a small 3D printed model of the uterus, because we do a lot of applications towards reproductive tract. Or let's say we do some urinary tract models where we sort of 3D print the anatomy, but also bring the biological part in, where we also eventually really 3D print with cell-laden ink. So really have a biological in vitro model there. So it does fit in. And I think important is also as postdoc to go a little bit out of the comfort zone and learn different techniques, so that you just add to your skill set, and you're able to draw from different fields, knowledge from different fields.
Yeah, I think it's a widening of perspective.
Yeah. Yes. Yeah.
That sort of approach where you spend some time to explore and see what's out there. and work on different projects. Coming back to microrobots, one of the applications that are kind of obvious when you think about them is treatment of, for instance, cancers. Can you explain to our listeners more or less how that would work?
Yeah.
How do we go from a sperm cell with an attached tube to actually delivering a drug to a tumor?
Yeah. So let's start with where we're currently at with cancer treatment, okay? Because the most common pathway is chemotherapy and radiation, still as of now. Chemotherapy means oral administration, ORV, and it's systemic, right? It goes through your whole body. It has a lot of side effects because of that, right? It does target the cancer, the tumor, the cancer cells, but also affects your body in many different ways. And that's why we still don't have a very good success rate with that, and due to the side effects, often the treatment fails. Same with radiation. The idea of using microrobots is mostly to make the treatment more targeted and active. So there's two things to it. On the one side, we have that active transport of our drugs or of a specific drug load, and you can use that many different propulsion systems for it. Okay, it doesn't have to be a sperm cell. It could be a magnetic field. It could be acoustic fields. It could be bacteria. Whatever you can to move something in the very complex physiological environment that we have. So the active transport is the one thing that overcomes, again, that systemic distribution of the drug throughout the body. And the second part is the targeting, to really know where the cancer cells are or where the tumor is and bring the drug load there. So it's a combination of active transport, precision, control. So control is very important in microrobotics, which again is linked to imaging, because we can only control them precisely if we know where they are and where they're supposed to go. And then having that target, and then finally having the targeted release of the drug as well. So this is in a nutshell what microrobots or nanorobots are hoping to do. And that will hopefully get rid of a lot of the side effects, and also making the overall drug dosage much lower, because if you're not distributing the drug throughout the body, but have it very, very targeted, delivered very targetedly, you can lower the overall dosage. And that should be a much better treatment option.
Did you have any proof of concept runs or any simulated runs where you've actually tested this out? Is this a very tangible thing in the very near future, or will this technology take some time to actually become common as much as chemotherapy?
Mm-hmm. Yeah, so because it's such a novel technology and we have a lot of new materials involved, a lot of new control technologies, imaging, so there's a lot of components to it that are new. It's not so easy to translate. Overall, I would say the technology, let's say in general, drug delivery by microrobots is right now in preclinical testing. So we do a lot of in vivo, ex vivo experiments, and again in different organs. So you can also imagine that wherever you want to deliver the drug to, so wherever the cancer is, you might need a different type of robot that is able to overcome, let's say, different biological barriers, or is imaged to a different penetration depth in the body, and all those kind of things. So now we enter that era, I would say, where we really have to tailor the microrobots to specific applications. Where, let's say, 10 years ago, the field was mostly focusing on how can we move it, like in general, motion mechanisms. Then how can we image them? And then came, how can we load them? And so now it's starting to really narrow down to the specific applications, and that's what people are working on. And not just me, of course, there's a whole community around the globe working on this. In our lab specifically, we're not that close yet to translation, we're mostly in the stage of in vitro experiments where we're looking at the performance of the robots, the fabrication of them, making sure we can image them, we can precisely control them, load them with drugs, kill cancer cells in vitro, and then when that's all checked, we are going to move to the in vivo stage.
Would you say then that of all the microrobots that you have developed so far, sperm cells are actually the optimal platform? One worry that I would have personally with something like a sperm cell being used as a carrier, as a microrobot, is how do we, for example, reconcile that with injecting something like a sperm cell into a bloodstream, right? It does not really necessarily belong in the bloodstream, especially if it's motile. Like, how would it behave?
Mm-hmm. Yeah, good question. I don't think there's any optimal microrobot. So, like I mentioned, they're starting to tailor them for certain applications. I would say the sperm cell might be a good option if we think about applications in the female reproductive tract, because they are kind of naturally designed to swim well in those fluids under those conditions. That's where I see the most potential. I wouldn't say that it has a lot of potential in other areas of the body. And also there are maybe other approaches that would work better. So we have some magnetic-based robots, or there are some that use bacteria-based robots. Again, there I'm not sure how well it would work. One big area that we haven't checked at all, and I think that applies to across the field, is the immune response as well. Yeah. So how quickly are the nanorobots or microrobots cleared from the bloodstream or from different tissue and organs? So that's also a big question mark. So
I see. That's also going to be something that needs to be really validated, because if we inject somebody and they end up having a very strong immunological response to it, it might end very badly. But yeah, I'm really glad to hear that we're getting there, that we're progressing from just in vitro studies to actually testing that stuff in real cells, in real systems, biological systems. So what does it take to take something that is already a living cell and tame it into something that does our bidding? How do you actually manufacture these microrobots out of sperm cells?
Just what I want to add on top of that is basically, what are these microrobots exactly made of? What is the material, the biomaterial that you use? And why did you choose that specifically, if that is something that can be told?
Yeah, we actually use right now we use very simple fabrication techniques. We've actually moved away from the microtube approach to just attaching magnetic nanoparticles to sperm cells and then use magnetic rotating fields to actuate them. So we're not dependent on the sperm's viability or motility. We have full control over the motion, direction, speed by the application of the rotating magnetic fields. That works well because they have a much longer life, shelf life. Mm-hmm. So we can just have that self-assembled actually. The nanoparticles and our sperm cells, they have different charges and they have electrostatic interactions, and we don't actually do anything. Yeah, they self-assemble and then we select the ones that work well, and we can use rotating magnetic fields to move them and then load them with drugs as well. So even if the sperm cells are not alive, we can use their, let's say, the cell body, which has the cell membrane surrounded, to just load drugs by physical absorption. That's what we do currently, but we're also hoping to make this more sophisticated and have more control over the release of the drugs.
So we were talking about microrobots, but also when I was researching the topic, I looked at it, and there were different terms for those small-scale robots. And one thing that may be confusing to our listeners is when we say microrobot, nanorobot, small-scale robot. Could you explain what are the differences on the order of size and how tiny these things are that you're working with?
Yeah. So we do work with a range of robot sizes. The smallest ones would be microrobots, because at least one length, let's say, of the dimension of the robot is in a micrometer scale. With the example of the sperm cell, we use that. A sperm cell is about 60, 70 microns in length, and the head is 5 by 10 microns. So they're definitely considered a microrobot. Then we have some that are more in the millimeter scale. So they are 3 to 5, 6 millimeters long, that we use for other applications like a non-invasive surgery, or we want to deliver a larger amount of drug or a larger amount of cells, then those would come into play. So these would be considered small-scale robots, technically or strictly speaking, not microrobots.
I see. So application-wise, it's treatment, like we said. Also, what seems to be often mentioned in your papers is the question of infertility. You also mentioned that the sperm cells could be somewhat naturally used in a female reproductive tract because they seem to be motile there in the first place. What approaches exactly do you have in mind when addressing infertility? And I think we could treat it under both angles. Like, can we address male infertility this way and also female infertility? Like, what would be the angle?
Yeah. So when we initially started with this work, our motivation was to develop a new assisted fertilization technique, where we have a way to actively deliver sperm cells to the fertilization site and thereby increase fertilization outcome. So basically take in vitro fertilization and bring it back a little bit more into the natural environment, because then we could circumvent oocyte retrieval, which is one of the big factors now in vitro fertilization. You first have to extract the oocytes, then you have to treat them, you have to mature them in vitro, then bring them together with the sperm cells, do the in vitro fertilization, and once the blastocystis forms, it would be transferred back into the uterus. That's currently the process. And our idea was, well, if we could just get rid of the oocyte retrieval, we make it a lot less invasive and also provide that more natural fertilization environment. Why is that important? First of all, it's very invasive, especially for the female patient. Second is there are a lot of very important selection mechanisms happening in the female reproductive tract that somehow render the most fertile sperm to end up fertilizing the egg. So we don't actually understand yet how that happens and why it's that one sperm. So we can't really mimic that in vitro. And another interesting thing is that in vitro, we're not actually able to fertilize an egg with a single sperm unless you inject it into the egg. That's called ICSI, intracytoplasmic sperm injection. But if you were just to put an oocyte there and put a motile sperm cell there, it would not lead to fertilization. And that's also something we learned along the way, because we thought, well, if we can just bring our one sperm cell with this little microtube to the egg cell, and then fertilization can happen, right? And we realized, no, there's actually so much else happening that in vitro you need thousands, if not tens or hundreds of thousands of sperm to fertilize an egg. So that's how it happens in vitro. So our whole idea of like just bringing one or very few sperm to the egg has not been successful yet, which has not to do so much with the fact that maybe there's something wrong on the microrobotics side, but more that we don't really understand the biology behind fertilization, that it's actually, you could say it's a group effort. There's a lot of sperm cells that play different roles in removing the cumulus layer. That's a small area of small protecting cells around the oocyte that have to be removed before the fusion can actually happen, and so on. So there's a lot of parameters that we, as let's say engineers, roboticists, did not think about. And now we're learning along the way and realize, oh, okay, maybe we need to change our approach. But then going back to the second part of your question of maybe what are other potential applications in infertility is I'm hoping that we can also use these microrobotic tools to study infertility more, and female as well as male. So a lot of times when there is infertility on the female side, there is some sort of pathology. Sometimes the anatomy is a little abnormal, or there are some obstructions, like the fallopian tubes are blocked, or other things that we can't even diagnose. So right now we have about 30% of cases of infertility that are just have unknown reasons. So the doctors don't find anything wrong, the patients are healthy, and yet they cannot conceive. So it also shows us that we have very limited diagnostic tools that really allow us to study sperm migration and all the events leading up to fertilization in vivo. Yeah, obviously it's a very sensitive environment, so it's very limited what kind of in vivo studies we can do. So the idea is that if we can develop tools that help us understand how sperm cells migrate through the female reproductive tract, how long they stay in different areas, how they interact with the tissue, how they stay alive, and so on, then maybe we can also treat infertility or give some implications on diagnostics and treatment.
You're very much on point with the complexity of embryology. Last time I was in University of Warsaw in my embryology classes, we did not really understand how acrosomal reaction happens. So that's for context. That's the sperm cell actually penetrating the membrane of an oocyte. So the crux of fertilization is that reaction. And at least last time I checked, we did not really understand how that process actually occurs, what triggers it, and what selection process is also done at that level. We just don't know. So the stuff around treating infertility is definitely way more complex if we don't really understand the mechanisms behind the fertilization itself.
Yeah, it's actually amazing that that in vitro fertilization works at all, and that it works so well. So with the fact that we can inject a sperm cell, no matter if it's motile or not, and we get 30-40% fertilization rate, it's pretty amazing because we don't actually understand how it naturally works. So yeah.
That kind of like makes me wonder if at all you do things at the micro scale or at the nano scale, to say the least, a scale that is basically just really small than what you see in real life. What are some of the other observations that you probably had along the way that actually differs from your hypothesis, say, at the regular scale that humans perceive things? For example, we probably thought that, like you said, taking the most strongest and motile sperm might actually work out in fertilization, but then it did not. So were there any other such observations on the micro or the nano scale when you get into those stuff?
Probably a lot of little ones. Yeah. I don't know if I remember any, but
something interesting that might have.
Yeah, trying to think. I mean, I did also some studies on kind of metabolics of sperm and kinetics, and I think that raised a lot of questions that I don't know if we really found a good answer to that yet. But like that example that you gave on right now, in the clinics, they would always select the most motile sperm because that seems to be the most healthy one. But truly in vivo, we don't know if that's what's happening because, sperm cells stay in the reproductive tract for quite a while. So it's maybe actually more related to being very efficient with the energy and say having a good energy saving mechanism that makes you basically stay alive for longest, or being more, let's say, the marathon runner versus the sprinter. Because if the cells use up their energy too fast, they might not be alive long enough to fuse with the egg. Other than that, I mean, there's a lot of not mystery, but a lot of tricky things also around motion on the microscale. So I don't know if you're familiar with that, but in general, whenever we think of robots, and their microscopic function, and we want to adapt that to the microscale, most of the time doesn't work because the forces and the physics on a microscale are so different. So that was kind of an eye-opener for me too, as a PhD student, just understanding that viscous forces and inertia and a lot of things are just so different from the macroscale. So any of the robots that we have in large scale, or even machines, and we think about how can we downscale them, you have to think of completely different approaches if you want to make it work.
So you're not essentially just a biologist at this point. You're an engineer, you're a roboticist, and what else?
I don't know. I don't know what I am. I mean, that's the thing, about working interdisciplinary. In the end, it feels like you're not fitting in anywhere. And that was actually one of the challenges when I was starting to apply for faculty position. In biology, they would say, you're not a biologist. And in chemistry, they would say, you're not a chemist. And engineering, I'm not an engineer either. So I think systems design is a pretty good home because it's a quite interdisciplinary field. So I have no answer to that. Yeah,
that's exactly why we are also part of the system design, because we have our undergrads in like completely different fields, and what we work on currently is computer vision, and that's completely different. Yeah. I will come to this question in the later part of the conversation when we have, but then I do have some questions with respect to how beneficial multidisciplinary approaches are.
Are you both in systems design here as grad students? Yeah,
masters.
Yeah, nice.
We're both working on artificial intelligence, but our backgrounds are in something else. So
biotech.
I'm biotech.
Electronic.
Okay. Yeah. Nice.
And part of the reason why I entered systems design and started learning about AI is basically sort of congruent with developing better cancer treatments. Because my imagination is that the only way to model complex systems like cancers, like tumors, is going to be pretty challenging if we don't apply some form of statistical learning to it.
Right.
And then in that spirit, I would probably want to ask you, what do you believe is the right approach towards treating cancers? You're mostly in the microrobotics field. The targeted delivery systems are absolutely awesome. If we can figure that out, that would be an amazing progress. But then there's also the question of, for instance, tumor heterogeneity. At some points, some of the tumor cells can stop responding to certain cytostatic drugs. What do you think the next sort of 10 years would bring in the area of cancer research, and what would be the best path to focus on?
Yeah. I think one big area that has a lot of potential is immunotherapy. So using patient-derived immune cells. So you basically would take some immune cells from the blood of the patient and amplify them and inject them back into the body to help the body attack or treat its own cancer cells, right? So I think that has huge potential. I've seen some cases where everything else failed, chemotherapy, radiation, and completely metastasized body, and then immunotherapy was able to save this person. So I see a lot of potential there. I think it's sort of similar to the topic of infertility in a way that as long as we don't really understand how cancer occurs, it's really hard to treat it, right? And treat it at the root. So I think it's a little dependent on that as well, how much more we are able to understand what causes cancer, what are the mechanisms in our body to really interfere with those pathways.
This is extremely tough because cancers are essentially a group of diseases, right?
Oh,
so
yeah.
Every cancer can be completely different.
Exactly.
And that's why I think you're very I think immunotherapy is actually the way to go. I think you're very right on that part because it allows us to adapt towards specific cancer cells. Do you think there would be a way to combine that with microrobots? Is this useful, for instance, to facilitate delivery of custom antibodies towards tumors?
Yeah. So that would be one thing one could try out, to deliver the immune cells or deliver the antibodies to specific tumor regions. That could be definitely a good way to go.
Nice. So I guess we could transition to maybe lighter topics because we're talking about cancers, and my grandpa passed away. He was diagnosed with leukemia when I was little, and he passed away when I was 4. So there's some memories that are getting stirred up.
I think almost everybody has these connections, right? Because cancer is so
pervasive. Yeah, it's super pervasive.
It was also for me one of the motivations. I went into biotechnology when my younger brother had leukemia when he was 2 years old, and I was 10 at that time. I think it was around that time, and it had a huge impact on our family. I was glad that he got through it. It was very rough doing chemo and radiation and all, but just seeing like a small child suffer for years and years through the therapies made me think, okay, there must be a better way to treat cancer. So it was definitely one of the things that motivated me also to go in biotech.
And how much in general do you think the medical community is actually receptive of technologies like these? For example, microrobots that you can basically use for either fertilization or for cancer therapy or stuff like that. How much does it take to actually bring it from the lab to the hospital?
Mm-hmm. Yeah, I think it's still going to be a long ways for the nano microrobots. Yeah, because it's not just a new drug or a new material. It's a combination of all those things. Yeah.
What do you think are the impediments to get there? Yeah. And what do you think is probably the most visible or the right path that one can follow to get there?
Mm-hmm. Yeah, so we need to have full control over deployment and retrieval of the robots. This is one big aspect. If we want to get this approved, right, we have to know exactly where the robots are going. How do we get them in and out of the body safely, right? Or if we leave them in the body, can we be sure they are cleared from the body eventually? There's nothing accumulating anywhere in another tissue. So that's similar to topics that we face with nanoparticles, nanomaterials, and so on. Another big milestone would be the imaging and precision control. So ideally, you want to have real-time imaging and control over the motion of the microrobots. So that's something a lot of people are working on as well. And sometimes the medical imaging modalities, like the standard ones, are not enough, right? Like ultrasound only gives us a resolution of maybe a couple hundred microns in good penetration depth. So maybe we need to use MRI, and then you need magnetic materials and so on. So that's one other big thing that we have to figure out. And I think those are mostly the areas that we have to adapt to human size as well. So right now there are some studies in rodents, but of course it's different if you have a magnetic control setup for a mouse versus for whole human body. So those are some of the...
I see. And one very interesting topic apart from all this is basically where you seek inspiration for whatever you do at this point from nature. And that's also been one of your core philosophies or your research interests and stuff like that. Can you just elaborate on what you mean by that and probably give us instances where you think there is actually analogy between nature and what you do, and how it basically just inspires you to do?
Mm-hmm. That's a lot of questions.
I know. Take your time.
So maybe talk about bioinspiration first. So bioinspiration for me means that really like going into nature and looking at how things work can be on all scales. For me, it's on a micro scale, but like even on big scales. And learning about how things work, how things were made, how things are fabricated, how nature kind of evolves things. So a couple of examples. One, of course, sperm cells we already talked about. So these are just amazing microswimmers that work really well on the micro scale. Yeah, they can swim in high viscosities. They're able to respond to all kinds of stimuli, surfaces, molecules like do chemotaxis, pH gradients, temperature gradients, and so on. So they're really smart cells. How can we learn from that to create microrobots that are able to do the same? Like artificial robots that are able to respond to pH, to temperature, to viscosity, magnetic fields, and so on, in a way that we have full control over the system and can use this responsiveness in a way to have control over their motion, their speed, and direction. That's one area of bioinspiration. Another one is for the fabrication. And before we started using the sperm cells as templates for microrobots by attaching these nanoparticles to them, you could also see that people would look at, for example, at plant structures, plant vessels, and you could find some really neat helical microstructures in some plant vessels and just isolate them and use them as a template for helical microrobots, for example, that are also driven with magnetic fields. So there's a lot of structures that we could just take advantage of. And that would be much harder or more expensive to build with nanofabrication, like with synthetic means. Yeah. Otherwise, for bioinspiration, I think it's more the idea of motion mechanisms, bacteria and sperm cells that can inspire us to build also synthetic robots. So we do a similar way, sort of look at like studying sperm cells, but then also fabricating synthetic, soft, flexible swimmers that mimic that motion, but are completely synthetic. So that would be another example of bioinspiration.
I see, because this question is of particular interest to us because we usually work in artificial intelligence, and the artificial neurons that you have here is basically compared with the neurons that is present in your brain. As much as you have your dendrites, your axons, and everything is essentially what has, in one literature, in one section of people who believe in this literature, these neural networks have actually been inspired from that. But there is always a congruent and opposite argument that basically tells that when you have systems that are designed for things probably outside of, let's say, something that is artificial, something that is not real, that is created by humans, you need not necessarily emulate what is present in the nature, because nature might have its own way of optimizing for things, and you might not understand what it is to the fullest extent. So develop things that actually works out for you. You don't have to trace inspiration from nature. It's kind of like one of the other arguments, and both these arguments exist in the field of AI. What do you think about these? You don't have to comment about AI per se, but you can think about if this argument is actually presented in your field.
Yeah.
What would you think?
I think it depends on what you want to do and how you get there. Like if you can get there without inspiration, bioinspiration, like it's completely fine. One example for that is, so we've compared motile sperm cells with, so they are just swimming on their own. We don't do anything to them, and we record their motion. And we look at their velocity, we look at their wave parameters, and so on. And then we take a non-motile sperm cell, we put magnetic particles on its head, and we rotate it. We create a motion that almost looks like a real live sperm, okay? So in terms of the wave parameters, it's quite similar to the live sperm, but it's completely induced by the magnetic field, okay? And we found that even though the wave parameters are somewhat similar, you know, the frequency, the wavelength, the amplitude. But the power output, like the force output, which can be measured indirectly by velocity and so on, is much lower. So the live sperm, the real sperm, does a way better job in using that wave like bending wave motion, and transferring that force into a forward motion. So in short, our rotating actuated sperm is much slower. Why is that? We are sort of inspired by nature, but we still can't do it the same way that the sperm cell does it. Why? Is in a motile sperm, you have these molecular motors all along the tail, which means the force is not only generated in one area, but it's in tiny, 8 nanometer sequences. You have all these tiny motors linked to each other, synchronizing with each other, and doing this amazing, group work, let's say, to have an amazing power output. So somehow, I think we're just not able to reproduce that as well. So in that case, I don't know if that's a good analogy or not, but I think we're just not really understanding it maybe we do understand how the force is generated, but we can't replicate it with artificial means. There's a lot of different aspects to it too that I didn't mention. For example, the stiffness of the tail that is a gradient across the length and so on. So there's a lot of other aspects to it.
There's also mitochondria in live sperm cells, right? So they have their own supply of energy.
Yeah, that's right.
Here you also need to transfer the energy from the outside. Does that also mean that you lose some of that energy in the process, or?
I mean, the energy source is completely different, right? So on the one side, you have the metabolic processes, the mitochondria that are the powerhouse and generating the ATP, and then this ATP goes to all these molecular motors along the whole tail. Whereas in our magnetic actuation, yes, we have that external force, but it only acts on wherever the magnetic particles are and rotates them. So, just to ask back maybe around about your question, do you think those two theories are then also questioning if we need, let's say, if we're in need of inspiration in general, let's say human input, or not? Is that a different story?
So, do you mean to ask if we need inspiration from humans per se, or from nature?
From humans.
Okay. Again, as you told, I think there are just two groups, and I'm more actually in the middle, I would say. And that's the safest point to be in at this point, I guess, neither sides. Because as you told, there are probably certain processes that require you to trace inspiration from nature, because that's the most optimized thing that you can also understand. But on the other side, there might be certain situations where you do not understand something fully, and then that'll be just half inspired, and your system might not even actually work out. Yeah. So I think this is also a significant thing in macrorobotics, where people actually want to anthropomorphize. Yeah, I always get stuck with this word. Basically, just make the robot imitate the human and make it as much as humanly as possible. But on the other side, that might actually limit its utility to just what humans can do. And maybe a different structure. For example, I actually was very surprised to see these two. Have you watched Interstellar? I think everybody would have. It's a movie by Christopher Nolan, where basically they just do interstellar travel. So they have a couple of robots there. One is called TARS. The other one, I forgot its name. So the robot structure is like completely different. I can actually pull up an image and show that to you, probably. But they were basically designed for utility more than they are supposed to be designed for, let's say, just to mimic humans. Yeah. So I kind of like think those are kind of like the situations where you basically have something designed for efficiency.
Do you
I
have
can pull
the
it
image?
up.
Okay.
Meanwhile, yeah.
Yeah, sorry, I'm not familiar with it. I'm not.
I'm gonna
Yes.
show you the image.
It basically looks like a big box
Okay.
that is capable of like I don't know. It has weird limbs which also look like boxes, but it can change shape.
Looks like this.
So it's
Oh,
not
wow. Okay.
I can
entirely
show you-
anthropomorphic.
Very abstract, so to say.
Yeah, it can just speak like humans do. It can have a proper conversation, but the shape or the form is not exactly humanly at this point. And so that exactly, I think, is the conundrum to mimic or not to.
Right, right. Well, it's also tricky because we sometimes forget how dynamic the world is too, right? Like we're never done with, this is all we know, right? There's always improvement and there's always like a lot of dynamics, right? So being inspired by something might look like this one day, but then might look different next year already. Do you know what I mean?
Yeah.
Yes.
So even in nature, like some of the things just don't seem optimized, and yet they work somehow.
My favorite example is the giraffe and the vagus nerve of a giraffe, which goes all the way down from its brain, slips underneath the aorta, and goes back up. It's it's very inefficient. So I guess in some ways evolution has artifacts, right? So some of the byproducts of evolution are results of essentially the prior things, the stuff that we have observed before, and we couldn't really change them very fast. But I would be more leaning towards, maybe also given my background, I'd be more leaning towards your way of seeing things, where we should actually take a look at around ourselves and see what out there exists, because looking at biology means essentially we're looking at the advantage of hundreds of thousands of years of evolutions of different species, right? So we're leaving that out on the table if we're not looking at systems that already exist and study them in depth to a point where we actually understand how we could potentially replicate them. Yeah. That would be the hope. I guess coming back to clinical applications and biotechnology itself, and I was talking about cancer treatments. One of the things that I was wondering about yesterday when I was preparing for the podcast was, we're in the space of AI, so we're always thinking about computational biology, about how do we model things in silico. Do you think that it is possible at some point for your group to also develop like a digital twin of a sperm cell, or of literally any microrobot, where you have a system that can let you run experiments essentially in a computer without having to do much in the wet lab, unless you have also that stage where you're like actively testing something that you are pretty sure that is going to work in practice. Do you think that would be useful? And how far away from that sort of scenario are we, where we can model an entire cell, its microenvironment and everything around it, that would help your research?
Definitely, yeah, that would be amazing. Yeah, I was hoping to do something like that, and I've been tossing this idea around over the last 5 years or so. Definitely. So the thing is, that's out of my expertise, right? So I'd be super happy to collaborate on that, and I've brought this up to in a couple of different scenarios, and said it would be amazing if we could create a digital twin of our microrobots. In the case of the sperm cell, it would be very difficult because it would be so hard to find the right input parameters, because we understand so little about the reproductive tract. But let's say for microrobots in general, especially going towards applications, it would be really, really good having that digital twin. And even in the final, clinical application, to have the digital twin like running in, let's say, as we do the surgery or the treatment, that's, I think, is the way to go, definitely. So in some areas it might be easier to do. So maybe that's something we should explore in the future. For example, and we have some applications more in blood vessels for blood clot removal or in the urinary tract. And I think it's a little more accessible and it's a little easier to find data to create such a model. So yeah, that would be really, really good to understand the interactions of robot with the environment and how to design a control and imaging and all that. So definitely, yeah, good idea.
I'm looking forward to the day.
For your second PhD, or I guess you're doing your master's now.
Yeah,
yes.
So first PhD perhaps.
Maybe
Funnily
sometime
enough,
later.
I'm being convinced by my friends to stay and do a PhD, and I've been defending myself from that so far. But yeah, there's definitely U Waterloo has so many amazing opportunities, especially I would say, especially for people who are interdisciplinary, that I would recommend anybody to spend at least the 2 years and do a master's here. Yeah.
Exactly. So we've talked about so much things that could be successful, yeah, but then could not also be successful. So the work that you do is so niche that it has a lot of pitfalls, and it probably hasn't been explored as much because you're basically breaking rules, both A, in robotics, where you have different physics and things happening, and B, in biology, where you're actually stumbling upon things that go against your hypothesis. So in these kind of situations, and in general in research, pitfalls and, let's say, disappointments are really common because sometimes you just don't get to where you want it to be or where you want to be. So how do you handle those situations? How do you handle failures in general in your life in research and basically putting in your efforts but just not being able to get there?
Yeah, that's a good question too. I think sometimes it's about having that wisdom to know, when it's worth to repeat and have resilience and stick with it, and when it's wiser to just stop it and fold it and focus on something else. Because there are routes like I've encountered as well, where I've worked on something for a year or two and nothing came out of it. It was basically had to fold it. But then, so my strategy, I guess, is first of all, is that I never put all my eggs in one basket. So I always try to have 2, 3, 4, 5 projects going. And that way you always, you have maybe success in one area and failures in others. And I wouldn't even call them failures, but, just things are not working out for whatever reason. It's able to help you keep going, because it's so important that we have something that keeps us going and a drive. So, and we all live from those aha moments and eureka moments where it's like, wow, this is amazing and it's going well, but then it's easier to forget about all those times that it didn't work. So that's sort of one strategy. And it's always it's hard, right? It's hard to know when to stop, when to start, when to repeat, when to keep going. So it's a lot of resilience too. And maybe sometimes it's on my side, it's more ignorance and just being sort of stubborn. So not giving up on certain things, or I keep asking or questioning certain things, and people tell me, no, that's how it is, and I keep questioning, and then sometimes it ends up kind of opening doors, and you end up understanding things more. But otherwise, I don't have a big problem with things, especially, let's say, on the experimental side. I think you learn so much and we do a lot of experimental work, right? So we learn a lot along the way. And when we, I also try to encourage my students not to call it failures, but really just, you do experiments and they might not work, or they don't get the result you're hoping to, but you learn so much along the way. You improve your protocols, you keep trying different things until you see something working. So it's a long process that requires also a little bit of that mindset, of just, it's not about the success, but it's about the process and about figuring out how to get there. So it's a lot of curiosity driven and just experimental sort of driven work. But I do also get a lot of encouragement from, let's say, from the research community. So I do a lot of collaborative work, and I think the teamwork is really important too. That helps you go like further than you could go alone. So I think that's another important thing. When you're stuck with something, always make sure you get somebody else to look at it.
Mm-hmm.
Yeah, that's an interesting point that you made as to finding the very fine balance between exhibiting resilience and giving up. So is that something that one just develops over time? Is that kind of like a second-hand feeling when you basically look at a problem space and you start looking at it? You get this feeling that this might go somewhere, or you just have to show a bit of resilience towards that? Or is it something that usually one gets from comprehensively thinking about things beforehand?
I think it's a little bit of both, because there's some experiments where we really have no clue what's going to happen. And I like those because whatever you get is good. But then there's some where I can, like, sometimes a student comes to me and they say, oh, this is what I want to do, and this is what I think it's going to happen. I can already tell right away, there's no way this is going to work. So it's a little bit of both, right? So you need to develop that fundamental knowledge a little bit, for example, about motion on the microscale, where you know certain things are just not possible. Without inertia, for example. And then there's the other part that is maybe comes with experience, where you have done this so many times in the lab, it should work. It's just a matter of getting it right and doing it over and over again. So it's a fine line. So some of it is experience, and some of it is being stubborn, and some of it is the fundamental.
It's nice. It must be a little bit of both, also in my view. Cannot be...
But I think the level of both is basically dictated through whatever that is. You probably need to have some time, because balancing both, it's exactly the big deal.
I think the time makes you learn.
Yeah, and what I would say, like, I've also done many mistakes in that regard, right? I looking back, there are some projects where I wish I would have folded them a year earlier and not struggled with them for another year, but there was nobody that helped me or told me, it's not worth it. So, and then the other way too, there's some that I wished I would have done, and I've never had the time to go down that road. So it's a learning process.
You get one, you give one.
Yeah.
I think that's very common in research, that we just don't know. Like, we're heading out on journeys that nobody has ever been on before, which is extremely exciting, but it's basically like Christopher Columbus going to America. Sometimes you might, you're not guaranteed that you're not going to sink along the way, right? So yeah, I'm sorry about that. Yeah, that was a very, very pessimistic analogy there.
You're in America.
Actually, yes. So I've already arrived, I guess. My question would be then, what motivates you? Like, of all the things, like, if you had to pick out, I don't know, 2 or 3 things in life that drive you forward, that even like in those tougher moments where some things are not working out, what gets you out of bed? Because every student, every grad student at least, in probably U Waterloo, and not only U Waterloo, had this experience of like, oh my goodness, my project is not working out. My supervisor is going to absolutely decimate me. How do I even talk to them? How do you get up? How do you keep on moving? What is the driving force?
That's a big question.
That's a deep question.
Yeah, I don't really think about it too much. A lot of it is the curiosity. I think that brings me back to, having thinking about problems over and over again, and just having that drive to finding a solution, or progressing and advancing knowledge, but also technology. So it's really that curiosity-driven research that I get to do. So, I think I love it even more now that I can give the direction to it 100%, that I have that freedom of exploring the different areas. So I would say it's mostly that curiosity. And then on the other side, it's that idea that I hope I can contribute to advancement for health technologies, for robotics, even if it's just a tiny, tiny little thing that will matter maybe after I'm dead, who knows? But I think that is something that I don't really think about, but I'm hoping that we are able to contribute to making human life better eventually. So
that's pretty amazing, because it's somewhat similar to my philosophy. It's if you can push the needle a little bit forward, if you can contribute the tiny amount, it's already good enough. And also that curiosity is a big driver. Like, what is out there? What can we still learn, and how can it help people? It's basically the biggest question. So you've been here over the past 2 years. You have also taught courses at TU Waterloo. My question would be then, what qualities do you see in students that succeed in courses versus students that succeed in research? Are these qualities somewhat congruent, or are there different strategies that people take and end up being successful? Because for our listeners, if you're doing grad schools, for instance, here in TU Waterloo, you have 4 courses that you have to take, but you do research on top of it. So it's basically you try to juggle both. In your view, what were the best qualities for both categories?
Yeah. So I taught undergrad and graduate course. Maybe let's look at the grad course first, because I think it aligns a little bit more between the students that succeed in the course, which probably are also going to be successful researchers. Because in the grad course, or at least the one I taught, we do a lot of paper discussion and discussion around different topics on biocompatibility of materials. And one of the deliverables is actually a research proposal. So it's very much about thinking in that whole realm of a research project, from conceiving the idea, developing objectives, the methods, all the way to the budget and how much time you're going to need for that. And I think that is, if you're good at that, you're also going to be a really good researcher and being able to be proactive in your project, and all the way to being a PI and taking care of that. So there, I think there's a lot of alignment between if you do well in the class, in the discussions, and preparing a proposal, and communicating your idea, and thinking about the methods that you need for that. That aligns quite well. On the undergrad level, I've only taught one course so far, and that's a first-year course, where it's mostly focused on the science and engineering fundamentals. So there, it's a little more difficult to stretch it all the way to seeing how this applies in research. So there, it's also a little harder to work on those skills already. But we try to do a little more hands-on and more applied assignments, let's say. And there I'm hoping it's useful. But otherwise, I've also had the experience that, for my time back in Dresden when I was a postdoc, I had an amazing undergrad working in my lab. He ended up being co-author of 2 or 3 papers from I think he was just research assistant for maybe 10 hours a week for about a year. He did incredible work in the lab, hands-on, absolutely amazing. But he failed a lot of his undergrad courses. He just wasn't good at those deliverables that they were asking for. And actually, we made a case. We were trying to get him pass, and said, look at these amazing papers. Those were not just conference papers or low impact. It was really, really like high impact, amazing work he'd done, and they said, no, sorry, he doesn't make it in the undergrad courses. And it was really showed me that you need actually different skill set to be good at research versus being a good undergrad student. So that was unfortunate. But yeah, showed me that there's unfortunately that discrepancy sometimes of what's asked for in coursework versus what you need in research.
Yeah. So talking about these rather characteristics for success in students, you have your lab. You have a lab, and what are the ideal qualities that you look at prospective students who are coming to your lab or who reach out to you? What are the key metrics that either quantitatively or qualitatively that they should exhibit for your consideration?
So we use quite specific methods that I know I cannot expect anyone to come in with, or very, very little, but it helps if somebody has some hands-on wet lab skills, know how to pipette, know how to do measurements, how to work with the microscope. Those are assets, but it's not key things that I'm asking for. What I'm usually looking for is the interest in engaging in our research. And having interest in it. I think that's the main thing, and it's hard to gauge. So I'm trying to get to know people a little bit, but yeah, really mostly looking for that interest, and also the ability to work proactively. So really take charge of a project and saying, this is my thing, and I want to drive this forward, because it's very hard for me to tell someone exactly every little step along the way. I need the people to think forward and think, okay, this is where I'm going. This is, I think, the steps I need to take to get there, and to proactively just go and do it. And then, of course, ask for help along the way. So those are some of the things I'm looking for. Another area is sort of the critical thinking and analytical thinking. That's really important. So whenever you get a data set, to really look at it critically, is this helping me? What is it telling me? What are maybe the things that didn't go so well? What is actually happening here? So that critical thinking, which is really hard to teach, but of course it also grows as you work in the lab and gain more data sets. I think that's mostly it. Yeah, and communication is also very important to me. So I want the students to engage with me, with their team, communicate their work on a regular basis. So that's another point that's very important.
Yeah, just gonna throw in a fun question right now. If you tomorrow, let's say, if you get a grant for $100 million and you do not have any stipulations on what you can work on, what would you choose to work on?
Yeah, it would probably, that's a lot of money.
I know. So you can also choose to not work on things. You can also choose to just just retire. Because there's nothing expected of you. Everything is just given in goodwill, and then you're just supposed to spend it the way you'd like it to.
Yeah, so I would definitely go, there are two main areas that motivate me. One is reproductive health, so anything related to infertility, but also cancer in the reproductive tract, endometriosis. There's so many diseases in the reproductive tract that we haven't even investigated very well. There's no treatment available. So that's a huge gap. And the other area would be cancer. So targeted cancer, delivery of cancer drugs, immunotherapy, and I think it would all have some sort of microrobotic component to it, most likely, just for the sake of having that active delivery for those two areas.
So that's basically a sign that you're on the path that you always.
Yeah, I do what I want to do. Yeah, that's true. Then I would probably do the digital twin, but I would have to hire people to do that, to bring that expertise in.
I'm about to check out and go into the industry for a couple of years, but on the offhand chance that I'm coming back.
Yeah, let me know. That'd be cool.
I'm signing up.
Awesome.
Okay, I think we can then move on to like more lighter questions, I believe.
Yeah.
Are they lighter? It's it's hard to gauge.
Yeah.
So the question that we really like to ask our guests is essentially what does your average day-to-day look like? So after you wake up, like what is the sequence of things every work day, let's say?
All right. So usually, I get up and I get the kids ready for school. That's sort of currently what I'm doing. So we're sharing the tasks. I get them ready in the morning. My husband is home when they get home from school. So we have breakfast together, get our lunches ready, pack, and then the kids go to the school bus, and I ride my bike to work. Thankfully, I just live 5 kilometers from campus, so it's a nice bike ride. And then really every day looks different, and that's what I love about it. So some days I head to the lab and work with the students. Maybe I have to do some training or I do some measurements myself. A lot of times it's a lot of time in the office, either preparing teaching, preparing coursework, or having meetings with collaborators. There's a lot of proposal writing going on, obviously, as a young faculty member, so trying to get larger grants. So there's some work on a bunch of papers we're trying to finish the manuscripts for, or they're under revision. Then there's quite a bit of service to do as well, and that again looks very different. So maybe some meetings throughout the day that are either online or in person, where I'm helping out in the program committee for biomedical engineering or hiring committees. What else? That's mostly it. I try to go for a lunch break with colleagues, just hang out in the lunchroom, and not as common here though as in previous places, but try to engage with people on a social level, let's say. And then what else? Did I forget something? Coursework, proposal writing, manuscript writing.
So how's life? Yeah, how's life in general in academia though? So for somebody who wants to venture into academia and who's probably just looking at this path, what is your lens of academia and then academic life?
From like a social perspective?
From your own perspective.
I guess
actually.
it would be both like social perspective and
Personal perspective.
how much workload it takes for you, because many people do who listen to the podcast are basically in the stage of deciding, are we going to go for grad school or are we going to go into the industry?
I think it's a very self-driven, let's say, lifestyle, because you can arrange your work schedule. Most of the time you can arrange it pretty well yourself, right? So you can say, okay, and in my case, I can plan the experiment whenever I want to, if I want to do it Monday morning or Friday evening or whatever, I have the freedom to do that. So there's a lot of freedom to it. I would also say it is quite family friendly. Mm-hmm. And I think it has it doesn't have that reputation necessarily, but it depends on how you create that space. And I think it can be very, very flexible and very family friendly if you're able to set it up that way. And that's what I found doing grad school too. Now, I did my grad school in Germany, so it might be a little different from Canada. But that's when we started a family, when I was a PhD student. And for us, it was the perfect time, because I only my project was dependent on me, right? I didn't have like a whole huge group being dependent on me as supervisor. And I was very flexible in terms of the work hours, and I had the support from the institute and from my partner and all that. So I think that's one of the things I like about it, that flexibility. I also love the international connections. So you get a lot of exposure to people from different countries, from different communities. Working with people across the globe is something I really enjoy. The traveling aspect as well, right? We get to go to conferences or give talks in different countries. So I really love connecting with people. And Yeah, and then I mean, on the other hand, it's not for everyone. So you need to decide if that's something you enjoy, like that sort of sometimes lack of schedule. So if people do well with it or don't, like it.
Do you like it?
I love it. Yeah. I guess I don't like when people tell me what to do.
Yeah, I think so.
So
You would prefer this over probably a job in industry or a corporate job.
I mean, there are also jobs in the industry that are, let's say, in R&D and that are probably very much self-driven and like curiosity-driven as well. I don't have experience with it, so I can't even judge that. But I do know that I would get bored if I had like if I would know exactly what I have to do in the next 3 months even, and that would be repetitive, I would not enjoy that as much as what I do now.
So when you're trying to figure out that work-life balance, so you have a family and then you have your job, do you use some sort of form of scheduling, or is this basically more of an ad hoc thing? Because I've tried many things before. I've tried calendars, to-do lists, and I seem to be returning to some combination of both. So like have a running to-do list of things that need to be done at some point, and then just pull across the things that need to be done on time into a calendar. But how do you, like, when you have a family, because also like I don't have my own family yet, is this sustainable, this type of scheduling, or do you think like the sort of more flexible approach towards calendars and scheduling meetings is more beneficial towards a good family life?
Yeah, I think if you have a family, you need the scheduling. Otherwise, it gets out of hand very quickly. Like even just to share the task, about like the kids have all these activities, so you have to make sure somebody's home when they get home from school, or they need a ride to this activity or that activity. So always make sure that somebody's in charge of those different aspects, as well as household tasks, right? Like simple things like who does the laundry, who cooks, and like just and we don't have a schedule for that, but we talk about it, and we sort of have everybody has their area that they're in charge of. In terms of scheduling, I think we're as a family, we're pretty spontaneous in a lot of ways. I know some people have a calendar planned out like for months. We're not that far ahead, but I try to know, be aware of what's happening this week. For example, I know, okay, what time do I have to be home, or is there anything special coming up? So I do use my Outlook calendar, because that's where everything goes in, all the work and my personal appointments as well. And then for to-do lists, that's evolving over time. So I used to do it on paper, because I'd be traveling, or
that's the best way, I guess.
like read a book and then just have a piece of paper and do my to-do list.
I do it the same way. A pocket notebook, and then I don't have unnecessary alarms blaring off. I can just tick it off.
Yeah. So I have that, and then you move things around too, right, over time. Like there's something high priority, and then something else new high priority comes in, so things get shifted around. But I do need that a little bit to help me. Okay, what has to be done, and how important are those things? So not only have a to-do list, but then also prioritize, and the whole time management part of it is very important.
Awesome. So along these lines, is there any one habit, or maybe even more than that has actually tangibly shaped you and your life? One habit that you would have probably observed in your own life, or you just believe, which can actually lead to professional and personal success, majorly professional success in research, and perhaps also in personal success. Something that you probably developed over time, or something that you see in somebody else.
I think that time management is a big part of it, and then I noticed that going into my PhD, because I did something else after my undergrad, and so we had a startup, and where you start having to manage your time, right? You don't have enough time to do all the things that need to get done, so you have to start really managing your time well, as well as the communication skills. So I remember going, like, basically starting my PhD with colleagues that had been fresh out of undergrad. And I just noticed I think that was one of the most valuable parts of being able to manage your time well and saying, okay, this is all the things I want to get done, but I have limited time. So how do I fit it all in, and how do I manage? And ending up maybe not doing everything 100% perfect, but getting a lot of things done to a satisfying level. And I think some of it is just naturally like you adapt to, especially once we had kids and I was in my PhD. It was like a lot of people were asking me, like, how do you get it all done, and how do you do it well? You can't get it all done, but you have to start managing your time well. And I think that you're just being forced into it. Like, it just happens as a mom and working in research. So I don't know, like looking back, I wonder, what did I do with all my time when I was a student and single, and now, having a family and a job and all that. But it works. You just adapt to it, and it's
time dilates as we look back.
I think there was a research paper that looked at this experience of time dilating as we get older, and apparently it's true. The experience is there. You mentioned a startup. Was that like something that you co-founded, or were you working at a startup?
So startup is, let's say, it's one way to say it. We basically started a farm. So my husband and I, both grew up on farms. So we're very close to nature, and we love working and gardening and growing food, as well as working with animals. So after we both finished school and we got married, we had so many questions to solve, like, because we were from 2 different continents, so we had to figure out, where do we want to live, what do we want to do. We were kind of tired of studying and sitting in offices, so let's say we had that curiosity of maybe we want to be farmers and we want to grow food. So we started, we had this startup, startup is maybe a weird word for it, but we started a farm, which means we rented or we leased land. Back then it was in Alberta, Saskatchewan, and we bought a milk cow, and we started growing vegetables and did direct marketing. So kind of like a market garden farm, so growing vegetables, having egg layers, so chicken and different small animals, and then would deliver to the local community in weekly boxes, as well as to local restaurants.
Yeah, I believe the lifestyle would have been
very different. Yeah.
Did you like it?
I did like it. It was a very good experience. It was very hard as well. So it was physically very hard because we didn't have a lot of machinery, so we did a lot by hand. At the same time, it was very rewarding. At each end of the day, you knew you had to work very hard because you could feel it in your muscles.
Would you go back to that lifestyle at some point?
Maybe when I'm retired, I'm not sure, or if it's possible to do both. There's something to it that's really rewarding, really healthy. I think growing your own food, and not only your own food, but so providing the community with healthy food is something very satisfying, and also physically, like, it's a very healthy thing. The part that I was missing was the mental challenges, I guess, that sort of drove me back into academia. But if I could have, the best of both worlds of doing both, that would be my dream.
So do you own a garden patch then?
I do have a garden patch, but it doesn't look that great yet. This is my first season trying it, in Waterloo.
That's going to get there, definitely. Okay, so I guess we're going to wrap up with this last question. And this is like a typical question that we ask our guests. What is the meaning of life to you? What is your personal philosophy? Was the one system that you try and follow and you would like to share with others?
Cool. Yeah, that's a very deep question. You guys ask very good questions. That's really hard to answer in a short sentence, but I would say, I mean, kind of also in line with the bio-inspiration and all. I think there is a purpose to life, and I think that there's also a reason why we're all so different from each other and so diverse. And I think the meaning is that we're all meant to bring different things to the table with the goal of serving each other and loving each other and in a way that only you can do. So whatever talents are put into you, I think there's a reason for them. And the purpose of that is to bring out the most potential of that and serve, let's say, the people around you to make this world a better place.
That's beautiful.
On that beautiful note, we hope you had a wonderful time.
Thanks, I did. It was lovely.
We sure did.
Oh, yes.
So,
Yeah.
thank
I've
you
been looking forward to this.
Yeah. Thank you so much for finding the time in a weekday and getting to talk with us. Thanks.
Thank you, too.
Thank you so much.